Welcome back to The Issue on the ASGCT Podcast Network. Today we have a very special episode on chemistry, manufacturing, and controls with our guest, Dr. Susan DaCosta, the Chief Technical and Commercial Officer at Genezen, a US-based CDMO for viral vectors. Fair warning here, we do end up geeking out quite a bit on the intricacies of defining your CMC strategy. So ready yourselves for a tour de force of analytical and process development for viral vectors. Do we cover all the highlights? Let's see here. Residual DNA, check. Empty, full, and partial capsids, check, check, check. Tailoring your preclinical and phase 1 strategy with your path to BLA in mind, check. But we also cover key questions like, when should CMC join a program team? And why your CEO should be having lunches with the analytical development team. Come join us as we hear about Susan's extensive career in this space and how she tries to help teams think through these topics today. The future of cell and gene therapy is nearly here. The ASGCT annual meeting is now less than a month away, and starting in a mere 6 days on April 22nd, you can get a sneak peek at the cutting-edge research being presented May 7th through 11th in Baltimore by downloading the abstracts collection. Explore the latest breakthroughs and discoveries shaping the field, and don't miss the premier event in cell and gene therapy. Visit annualmeeting.asgct.org to learn more and register today. See you there. Welcome, Susan DaCosta, to The Issue on the ASGCT Podcast Network. We're so happy to have you here today.
Thank you, Emily. Thank you for having me. I'm both humbled and honored to be part of The Issue.
Yeah, well, I'm glad to have you here because what we've been doing on The Issue is sort of walking through all the different functions that sort of come together to build these novel therapies one by one to try and get a bigger picture of everything that has to happen in order to bring these therapies to the patients that need them. And so you're here to walk us through and decrypt the mysteries of CMC, otherwise known as chemistry, manufacturing, and controls. But before we dive into that, can you tell us about your background?
Sure, thanks. So I'm a molecular virologist by training. I have spent my entire career working with viruses. From my first half of my career has been with basic research level in an academic setting. I worked as a research faculty at the College of Medicine at the University of Florida. The second half of my career has been in viral vector gene therapy drug development. And so a major chunk of my time in drug development has been with contract manufacturing and development organizations. So I started in 2013 at Florida Biologics when the company was still part of the University of Florida. And I've grown with the company during its evolution to Brammer Bio and then to Thermo Fisher Scientific. I've had the good fortune of spending and exploring every aspect of CMC as it relates to viral vector gene therapy. So I'm really fortunate to have had a very expanded view on how CMC looks like in various different aspects. So to begin with, I was responsible for quality control. And I spent a lot of my time there developing and qualifying viral vector analytics. And I like to say this because I think it's really important that my time in quality control really colored my science in a positive way. And I truly understood the value of you cannot unknow what you know. It's so relevant in the context of drug development and patient safety. And my time there was soon after Glybera was the first AAV that was approved in the EU in 2012. So it really helped to think about these assays and understand how we were uniquely poised to be trailblazers and develop and qualify these viral vector analytical assays. A predominant part of my career at Brammer has been in process and analytical development when Brammer was at its peak. And this time came with excellent learns, knowledge that was varied yet vast. So we worked with adeno-associated viruses, lenti and retroviruses, some HSV. So it was really great, and a lot of adenovirus as well. It was really great to be able to find all these varied conversation pieces pretty much in the context of understanding what different companies were doing and how they were really thinking about their CMC for drug development. And I have to honestly admit, I'm a little bit of a data junkie. So I loved being able to look at all these data, evaluate the trends of how these residuals appear in these different processes, and how the different platforms, when we're talking about AAV production, impacted these impurities as well as the efficacy and productivity. So I had a really good time understanding the regulatory landscape as well. And as you may recall, we're still nascent in our regulatory landscape on knowledge, but at that time it was really out there. And just understanding and kind of building out this context and how it impacts patient safety was really critical. So I think that was a great learn. And then as part of my role, I also was involved in tech transfer of process. So it's from development to clinic as well as commercial manufacturing. So this is when Brammer was really exploring the commercial manufacturing landscape as well. And I got a chance to deep dive into programs in late stage development, look at PAI inspections, kind of expand my repertoire of knowledge there as well. So I think all of these experiences truly helped me to become one of the technical SMEs for both clinical and commercial manufacturing. When I transitioned out of Thermo Fisher, I spent about 2.5 years working at a therapeutics company and as chief technology officer. This was just during the pandemic. I had the good fortune of continuing to work remote. So I stayed on in Florida and worked in the company that is Alcyon Therapeutics that was based in Boston. I was responsible for all of their CMC, both internal and external. And this was for several AAV9 assets, but they were part of a neurological domain in that context. The company itself had started off as a device development company, and it worked with devices that were exploring the ability to advance and biodistribute medicines in the neurological space. And then they decided to go expand into gene therapy, and really their flagship asset or clinical indication was Rett syndrome. And I truly got the ability to kind of explore how to build out the CMC for that, and then also think about a commercial device that is now currently been clinically validated, but then to be able to then explore a very novel way of thinking about fixing Rett syndrome using X reactivation and a novel way of distributing the viral vector in the deeper areas of the brain. So I kind of come full circle at that point. And about 10 months ago, I got reintroduced into the CDMO space. And this was as a result of a partnership that I had built while I was at Brammer Bio. Our chief financial officer at the time, Steve Favillaro, took on a role as CEO of a CDMO organization based in Indianapolis called Genezen. And Genezen had already built itself as a really bringing value to lentivirus and retrovirus gene therapy in terms of development and manufacturing, and really coming out of Indiana University and having really truly good technical experts there in the form of their chief scientific officer, Pratima Cherukuri. This was great. But when Steve joined, he thought about expanding the knowledge base to include a little bit of that magic we had at Brammer, kind of bring in AAV gene therapy, really do one of the simpler of the more complex viral vectors, right? And that's where he made me an offer I couldn't refuse. And the thought of going back again, creating, building, and really rekindling that passion, that knowledge, that technical expertise, taking the learns from what we had at Brammer and then rebuilding a new CDMO, or rather rebuilding or expanding the CDMO at Genezen was really something I was excited about. And that's how I ended back at Genezen as the Chief Technical and Commercial Officer here.
Yeah, no, that's an amazing career arc, right? And it's funny when I think back to my early days, just about a decade ago, first getting into gene and cell therapy. I think it's hard to today realize the impact that University of Florida, and what sort of grew out of that and became, Brammer in time, to underestimate how cutting edge you guys were in sort of setting the stage for everyone. That said, I think we all sort of have a sense that we've come an amazingly long way. For me, I remember it, or I think about it similar to, for a while I worked on enzyme replacement therapies, and there was it's funny, I came into that a little bit late when it was a very mature field, and we already knew about things like the level of mannose-6-phosphate on the enzyme made a difference in whether or not it got into cells. And so we knew that was something we had to look for. But analytical development around gene therapies, back in the day, you guys were still trying to figure out what were the key parts of the virus that you needed to assess. And so I'd love to hear a little bit about what you think the most impactful changes have been in your career. Like what sort of, I remember early days where on one project, our quantitation assay was like plus minus 50%. And we were like, great. Yeah. So obviously those things have improved, but like for you, what has made the biggest impact in sort of analyzing the virus and that quality control that you talked about in the beginning of your introduction?
Well, yeah, and you're right. In 2013, I think that's when I joined, the field was relatively young. It was, even though there was one approved AAV product, and there was definitely a lot of knowledge that came from Adno, Lenti, Retro, there was still not a good understanding of the regulatory landscape and what analytics should and should not be included. For example, even with the process, adherent transient transfection processes was the most viral vector production go-to, and that too in cell stacks. That was just the go-to for almost all of the viral vectors. I think Florida Biologics was fortunate because we were ahead of our times. We were one of the few select CDMOs that worked with baculovirus-based AAV systems, and we did use stir tank reactors that could scale very well to make the product. But like you said, the analytics were very nascent. We were still using vector genome titer assays that were based on relative quantification, not absolute quantification. We call them quantitative PCR or qPCR. And you're right, the CVs were enormous. The actual detector really only claimed to have a 50% accuracy at that time. So to say that your method was more accurate or more precise than that would not necessarily fit the conversation that's being had. But now that we have digital droplet PCR or droplet digital PCR and dPCR itself, I think we've come to an absolute quantification conversation. We've been able to drive those CVs to less than 15%, like the FDA requires us to. So we can truly compare doses, and we can keep those doses as close as possible without kind of overlapping what the true titer is when we're looking at the dose escalation experiments, right? So I think that's a very critical part. The second part was the process itself didn't require us to use a polishing step. When we did a downstream purification, we had capture followed by TFF. And so we did have a lot of empty particles, right? We're talking about a time when we were still in two camps and arguing on whether there was value to bring those empty capsids into a drug as part of the drug, more as a decoy system. And it took us to have several clinical trials and unfortunately a few deaths to truly align on the fact that it is important to enrich for our therapeutic DNA-containing AAV to get rid of as much empty capsid as possible, because especially in systemic doses where the payload is very high, and now you're adding a significant number of empty capsids, you're bringing in a significant amount of immunogenicity that we can avoid by enriching for them. We didn't have good technologies then to actually evaluate what the empty and the full capsids were. We still used electron microscopy, but now we have analytical ultracentrifugation, which is a gold standard that we normally use these days. But even that technology, we use too much. It's not QC friendly. It needs too much volume to be able to analyze the data. So more recent state-of-the-art technologies that use maybe mass photometry, maybe SEC, size exclusion chromatography with MALS and other detection systems that will help us have been really able to have really helped us to progress ourselves towards that area so that we can have a better understanding. Another very key analytical assay is sequencing. We didn't truly understand the quality of our transgene. Today, we have next-gen sequencing technologies that can go down to a single genome and help us to identify the quality of that transgene. Are our ITRs intact? Is within the transgene, are there small SNPs that we didn't realize that impact our potency, and how much of them are there? We can even use that technology to identify more impurities and potentially contaminants as well. So I feel like we're in a better place to be able to understand our viral vector product before we're able to put it into the patient.
You're absolutely right. I remember arm wrestling over empty versus full and the decoy hypothesis back in the day. And now it feels like, we've gone from thinking about this in terms of empty versus full, but also empty, full, partial, to your point, what percent have a SNP that might be deleterious, et cetera. And so our ability to much more granularly triangulate on what's in the vial has improved. The one thing we've been talking a lot about the DNA component, but obviously also you have the capsid itself, right? And do you also feel like we're learning a bit more about what makes a good capsid as well? Are there new techniques that we're using to figure out if the capsids are healthy capsids, so to speak?
Yeah, so I think initially when we first started working with these capsids, and there's so many different flavors of them, at least 12 natural serotypes, and so many of the serotypes that come from animals, come from engineering them in the laboratory. So we have all these various and sundry serotypes that we can then use to be able to, quote unquote, deliver our payload to the right region of the human patient that we're looking to go to. But it was not very clear in our early days about how these capsids are in terms of the quality of them, how many there are. During the process of purification, are we losing some level of potency because we can't transduce with these capsids? These capsids are falling apart. But now we have a better understanding. We do forced degradation studies with these vectors. We can actually degrade them on purpose and then look and see what that profile looks like. And with better technologies, we've got capillary electrophoresis, SDS, that we are using. We have better mass spectrometry methods that we can use to identify, analyze, and kind of understand these capsids better. I think we've kind of come again to the next generation. We can definitely look at them through EM. We can look at them through mass spectrometry. We have other technologies that we've been applying, and kind of you have better transduction assays, not just the tissue culture infectious dose 50 that really didn't tell us enough. We have better transduction assays that we can utilize to be able to characterize these capsids better. Because that vehicle is so critical for us to be able to take that therapeutic DNA or nucleic acid payload into the cell. And without the ability to transduce those cells, we could never bring that DNA to the right place. So you're absolutely right. And we've really kind of navigated that space as well.
Yeah, no, it's truly amazing. In your role, I would say probably both in a CDMO type role, but as well when you're in a company who's just jumping in to viral vectors for the first time, you're in the coach role, right? Of trying to help them think through viral vector manufacturing. And obviously in a short time, we just covered a lot of considerations and we only touched the surface of these things. So what are the key bits of wisdom that you impart to the teams that you're trying to coach through their first time thinking through producing these viral vectors at scale?
Yeah, I find this quite a loaded question because I don't want to diss another industry or field, because I mean every one of them are equally important and have provided really valuable medicines, right? But it is true to say that the complexity of the field has increased as we've gone from small molecule science and research to build those drugs to going into more complex molecules like biologics that are more monoclonal antibodies, but they are still a single macromolecule. And now we've gone into viral vectors, so to speak, that are just not one macromolecule. You have the therapeutic payload, which is a nucleic acid. You have the protein shell, which forms the capsid and is important for transduction. And oftentimes, when we're talking about lenti and retroviruses, we have the envelope, which is really critical to be able to maintain that potency as well. Yeah. So we've increased the complexity of our molecule, and understanding a molecule is really important. So one of the first things I would say to anybody that's new in this field, coming from a different field that has been exposed to drug development, is to understand your molecule. It's really important to understand it because the shear forces matter, how you're scaling up matters. You might be able to have kind of come up with an amazing process that you can use for biologics that you now no longer can use for viral vectors because it's too harsh. Even going from AAV to lenti, you use the same nuanced process that you're using for AAV, you might hurt your lenti because there are different things that cause the viruses to break apart or be fragile, right? So we have to really understand our molecule better. In addition, our QC is more complex. We're really talking about different quality control assays. They're not all HPLC, high throughput, fast. We use characterization methods a lot that we cannot quote unquote fully qualify, but still use them in our understanding of what our vectors are. So kind of getting that better understanding of what the critical quality attributes need to be, what the quality attributes in general are, and how we can make this more platform and less bespoke. That's important, but not everything can be platformed, as we've realized with working through this, right? And then again, the regulatory landscape is not prescriptive. It's still being developed. Even now, the FDA will not tell you that you need X percentage of full capsids, otherwise your product won't make it through and get approved in an IND. They will still want you to use the data from your animal studies and your toxicology studies to understand what that safety parameter looks like. Your residual host cell DNA, we know that these viruses are going to encapsulate a certain amount of that DNA, and you can't walk away from it. You can tweak that, you can make it better, you can hone in on it, you can decrease the amount of starting material that you're using so that you don't have to worry about this, but you can't get away from that conversation. So kind of understanding those concepts, figuring out what the trends are, and working through it. So my advice would be step back, absorb it all. Draw parallels where you can, because there always are parallels. Bring learns from your current industry where you were challenged in the past and how you navigated that. And I think those analytical approaches are all the same. But more importantly, think outside the box, like get disruptive. Don't try to just kind of check that box and fit it in. Just get disruptive, step back and say, well, this approach isn't working. What can we do differently? How can we think about this paradigm differently so that we can solve for it rather than say, why won't it force? Why can't we make it work in this conversation, right? So there is a lot of ambiguity that we are navigating, and we're going to have to kind of help each other understand that it's okay to navigate that ambiguity. And it will have to be a case by case in certain cases of our drug products that we're making to help understand the true meaning of what we're doing in terms of benefit to risk ratio, right?
Yeah, no, that resonates so strongly. I remember, so when I first joined, my first viral vector drug development team, you used the term ambiguity, and that is absolutely what we all had to get comfortable with. This was a group of folks who'd, they'd worked on small molecules, they'd worked on, some protein therapies, some enzyme therapies, and yet the level of ambiguity we all had to sort of hold hands and get comfortable with as we moved the program forward was immense. And I think, to your point, like we've had advances since that time. Luckily, there's a little less ambiguity today than there was back in that day. But there's so many unknowns, and it means that you have to sort of think strategically about how are you going to collect data in the clinic to be able to look back at the material you used in the clinic and try to, discern what qualities of those batches might be adjusting the benefit or the lack of benefit that you see as you sort of move forward in the clinic. And I would say there's a lot of science, but there's also a touch of art as well that teams have to bring to this.
Yep. A great example is the WHO recommendation of 10 nanograms of OCLD or of impurity DNA in
Yeah.
your dose. How is that even possible that you can put 10 nanograms of DNA in a dose when you're putting hundreds of milliliters sometimes of that dose systemically into a patient? It's just not a tenable approach. Yet people tend to get prescriptive and say, well, the guidance says 10 nanograms of DNA per dose is all the impurity you can afford to, and kind of get into a situation that is like very futile and say, we can't make that happen. And then when you talk to the FDA, you go through the pre-IND and you explain the data, you have your toxicology data, the FDA, of course, will come forward or any of the regulatory agencies will be able to help you out to understand that. And so I think as a CDMO, I've had plenty of examples of when we've had to talk a client off that cliff, off that edge, so to speak, and bring them back down and say, it is not an absolute prescriptive number. It has to be specific to your clinical material and what you've already determined as safe based on your toxicology studies.
Right. And yes, I've had that conversation on Teams more than once, that specific conversation. So I remember it strongly. And to be fair, like the WHO created that on the basis of other kinds of drugs, which are given more than once, whereas these, in theory, are single dose drugs. And so one does have to work with the regulators to figure out what makes sense, what your preclinical tox data substantiates for you, and navigate a path forward for sure. Let's talk maybe a little bit about the kinds of programs you've seen, because, I think you've probably seen it all at this point, from, high dose systemic viral vector therapies, and then moderate dose systemic therapies, and then, local administration, tiny doses, if you will. And from each of those different types of products, you're going to have different yield goals and probably also different sort of manufacturing and regulatory strategies, because when you go for, to get approval from the agency for a BLA, you need to have a certain amount of batch experience. But if you're, and you'll get that if you're working on a super high dose systemic therapy, because sometimes you may only have enough for one patient from a batch with all the testing that has to go on. But for some of these locally administered therapies, a single batch could get you through a lot of patients. So
Correct.
How do you help folks think through the spend that they do early on in a program while they're still trying to figure things out? How do you help them think through whether to spend at risk and at least make a second batch to make sure that the process is robust and that they're not going to run into troubles down the line as they, get closer to approval?
Yeah, that's a really important and a really good question. My time in the therapeutics company actually helped me to realize that there are huge disconnects between the preclinical, non-clinical drug discovery parts of the company versus the clinical development part of the company versus CMC. And CMC needs to take their take their knowledge, their baseline from both the non-clinical as well as the clinical development functions of the company. So my advice is to start at the target product profile. Like the company the clinical development team has a good understanding of how they want to deliver this drug. They may not have it down to a prescriptive mode, but they do have an understanding. So what is the incidence of the disease that you're looking at? How many total patients will need this medicine when you are in a commercial scope? Because that will really kind of help to navigate what kind of batch size you want. What target concentration you want, that will also determine what the batch size needs to be. How much total vector per patient, that is also another factor. Then what is the route of administration? Like you said, if it's systemic, then you can put hundreds of milliliters, but it also means that over time, you're using a lot more of a total vector genome dose as compared to something else. And then, if it's locally administered, how is it being administered? If it's an immune privileged area, you need to worry about maybe risk of impurities, like your product and your profile impurities, product and your process impurities, sorry. in the context of like residuals, but also in the context of empty capsids. It may not be as big a deal in certain cases, but it might be a bigger deal in other cases, right? And then the endotoxin levels, because if you are doing an infusion versus a bolus, how fast you're flowing that material through that organ is going to make a difference on how much of a shock that organ or that patient can feel when that endotoxin is there. So all of these numbers we need to understand better as CMC levels, because just even our raw materials will have a limit of detection endotoxin number that we need to be able to think about and bring in. Something as simple as Wi-Fi has a level of endotoxin which it passes muster and comes into the in the raw material approved and released stage into our GMP facilities. And so thinking of whether it's going in our brain or whether it's going in systemically will matter in terms of that endotoxin. So once we've kind of understood that, we can really then unravel that quality target product profile and understand, like, are we going to go after a certain modality in terms of the process? Like if it's AAV, do we need for a systemic conversation, you need a lot of vector. Should we go after a baculovirus system? Should we go after a scalable producer or packaging cell line? Should you just go transient transfection to begin with and then change your strategy later on? But that means that you now need to do comparability. You need to be able to then cross over, maybe not need to go into an animal, but at least analytical comparability is important. Right. What are the safety considerations when you're talking about these different processes, for example, right? Is the potency going to be different if you went into a baculovirus-based process versus an HEK-based process? What is your productivity and yield looking like? Because those are critical aspects, based on what that dose of impurities is coming on, what the safety considerations need to be in the context of how it's impacting your potency and just general safety. All of that needs to be kind of put in place. And then if you're truly planning for commercial process, you really need to think about, can I start at the scale where for clinical approaches, it's fine, and then I can expand up to a scale up rather than a scale out conversation, yet managing that conversation, Emily, that you pointed out, where the number of batches is important. So, and then when you have multiple assets in the same laboratory, in the same company, you're looking to find a streamline and kind of build a platform that can work for all of your different assets. So you might have an asset that requires a very low dose, or it needs it at a localized region, or you need moderate doses, and then something that has a much higher dose because you've got a huge incidence of patients. So if you can kind of build the platform so that your unit of operation can be a certain volume size, and you can scale up or scale down based on that unit of operation, you wouldn't have to have your CMC team go crazy, kind of validating this process and then validating another process, kind of come up with what would be the best approach to take. And also, if you can unify that platform, you can build a common reference standard, especially if it's the same serotype you're going after, that then gives you trending data across various different products, and you can actually use and navigate that more to your benefit. So when you go to the FDA to talk about process validation, you're actually being able to use that reference standard in the context of how this process is working for several different processes, several different products, right? Yeah. And then also the analytics, can we kind of customize, more platform the analytics with just a few assays being bespoke, and even those assays like the VG titer assay or maybe a potency assay being bespoke, can we find elements that we can platform around? I think those are kind of things that we need to think about as we're building out this process. So then when you go after rare and ultra rare and nano rare diseases, where it is just not commercially sustainable to build out a process validation approach, you can actually take this approach that multiple of these ultra rare diseases can actually make it to a commercial stage, because you're using more of the aspects of process validation overarching through different products, right? And I think Peter Marks talks about it a lot as well, about how he has a current pilot program too that will help with these rare and ultra rare diseases to be able to make it. So in short, I think the answer is you're never going to come to a good, happy medium, but if you can think about it more on the holistic approach for that particular company and what its assets are, so it can mix and match this and figure out how it can validate more of a platform approach rather than a bespoke approach for each of its assets, as long as it uses like common serotypes that it can use across the board. That would be the way to think about this.
Yeah, no, that makes a lot of sense. So, just thinking that through for a company that maybe has 3 assets, if all of those could be AAV9 because whatever you're focused on AAV9 type tissues that can be transduced by AAV9, you're in a better position to platformize quite a bit of what you're doing. If you for whatever reason, because of the different transduction patterns you're trying to optimize for across different programs, you're having to do one serotype versus the other. You're going to get less, I guess, crosstalk and benefit and cross comparison between your programs to sort of leverage as things go on. That makes a ton of sense. Well, and it occurs to me that sometimes, some of the teams I'm on very much start with a nucleus of researchers who are simply just trying to figure out, does gene replacement, for instance, work in this indication, right? And if you had your druthers, when do you think a CMC expert like yourself should join one of those research teams? Like, at what point would you like to be in the room so you can say, hey, if we just tweak this one thing a little bit, you guys are going to be much happier 2 years from now? Like, at what stage would you like to be in the room? From the beginning, or?
I think the stage I would say is the minute you discover that you can cure a mouse. Okay. That's when I think the CMC, at least somebody in the CMC team, should be weighing in on, now that you've figured out that you can cure a mouse, let's talk about how we're making your preps, even at the RUO level, at the research level. How are you making these preps so that you're not changing the kind of results, the kind of data you're bringing up? Because you have a pretty vast preclinical data set that you want to build together before you go and say, okay, this is ready to build out a company. But the minute you realize you can cure a mouse, I think that's the time to just open up those conversations and say, hey, is it possible to at least think about some aspects of it? Clearly, you can't figure out what your process development needs to look like at that point, but at least like some critical attributes that we can kind of talk through and say, these are the things that you want to kind of maintain. So we're still kind of seeing a level of quality of that vector going forward without changing the process dramatically as we move. Right?
Yeah.
I can't expect a research laboratory to have bioreactors, but shake flasks, you can always put a shake flask in there and make some suspension material, right? So kind of thinking more at that scale, I think would be important, and having some critical assays that will help to kind of understand what the empty ratios are, what the full ratios are. Maybe you use ultracentrifugation, which is a great technology in my opinion, and you get that really good high enriched capsid, and then use that information moving forward, right?
Yeah. Yeah, no, that makes sense. It's definitely one of those things where, well, I would say this, even at a minimum, if you're running like the study, the one that you're really excited to run, that you've seen some promising results in a couple of mice, but now this is the definitive study, save some vials for analysis of your drug product at a later point when you have your bona fide assays up and running. Okay. So that you can compare. It will feel painful at the time because for a lot of these assays, it takes a lot of volume to run them. So it may feel painful to set aside those vials, and may reduce the size of your study or make you run 2 studies. But frankly, there's a great deal of benefit for being able to look back at that material and confirm with your bona fide assays once they're developed. That what you thought you gave, you really gave. Yeah.
That analytical comparability will definitely go a long way to be able to then kind of merge and stitch all of the data together as you get bigger and bigger. Yeah, you're absolutely right.
Well, and let me ask you this about analytical comparability, because this is something I think a lot of teams face, and also I think about a lot. So as I understand the regulatory structure around drawing a comparability argument, you start sort of with analytical, then maybe you go to preclinical models as a next step if analytical is not checking all the boxes for you of comparability. And then, at the end, you may have to go into the clinic, right, and do a proper comparability in the clinic. And so
Correct.
I'm curious how far you think we've come on the analytical side in our analytical technologies. Do you find that more teams are able to satisfy that comparability argument analytically alone now? Or do you still think teams are having to leverage preclinical data or clinical data if they have a major process change, for instance, between what they did in phase 1 and what they're doing in their pivotal study?
Yeah, usually I think when a process starts to develop, it makes for better, more potent, more efficacious virus rather than less potent, less safe virus. So when you think about it, if your analytical approaches, if your assays are fit for use, if they have been qualified appropriately, and you can go back and take the material that you did, your mouse toxicology studies, your first early clinical, and now the new developed process, and kind of match it together and show that that scope is actually working, I think it should work. The time when it becomes a little difficult is now that you've improved, quote unquote, the efficacy. Now you're no longer in that same realm. So is that dose right? Is that
Right.
dose too much? And that's, I think, when the question comes up of saying we probably need to go back to the clinic to be able to show that this is no longer the same product. So we would have to go back to the clinic to show that the dose is still right. If we need to do some more dose escalation studies, this is the time to do it.
Yeah.
So more often than not, if I was the clinical developer, I would say, don't change the efficacy of that material. You've already proved the efficacy. Don't change that product in that context. Just focus on making sure that the process is, even if you're developing better, the process stays, the output, the final answer is the same. So we don't have to go there. I think more and more we find, at least in my experience, that the analytical comparability is enough and provides enough justification to the regulatory agency to move forward with it. But a lot of times I think this change will happen while the program is still in the clinic. And so you could kind of run two arms, the first arm being with the earlier product and the second arm being with the later product, and then match the two without having to spend an additional amount of time and money to kind of bring this out in a later stage.
To do a dedicated study.
But more often than not, I'd say the analytical comparability is enough, especially if the assays can show that the material is the same.
Yeah, no, that makes sense. And obviously, I think sort of embedded what you're saying there is the potency assay, which usually people sort of kick the can a little bit on. But that potency assay, the earlier you have it in place, the easier that analytical comparability is going to be. Correct. Because usually those assays sometimes it's not a single assay, sometimes it's a multi-part assay
it could be
with
matrix,
an aggregate.
Yes. Yeah. Correct. Yeah. Yes. And I think having some level of understanding of your potency as early during drug development as possible is really important. And you're right, kicking that can down the road is going to just hurt, not help, because now you don't have no understanding of how that efficacy was in vitro. And now you have to build it much later in the drug development life cycle, which definitely is not helpful.
Yeah, I think that if you're listening to the podcast and you're a first-time CEO, first-time gene therapy person, take your analytical development person to lunch with you. Like, go to lunch, have a conversation, have them teach you what's in their head and what's keeping them up at night. Because I do think that the better perspective we can have sort of at all levels of the organization about the complexities here and why it's important to invest early, even though it's it's not inexpensive work, but it pays off in the end.
You're absolutely right. And anecdotally, this is really funny that you said have as a CEO, take your analytical development person to lunch, because I just got off the phone yesterday with a CEO of a gene therapy company. And her question to me was, Susan, how much vector genomes did XYZ clinical trial have? And oh my gosh, that sounds so little as compared to what I'm trying to do for this neurological disease. And my answer to her was, you're comparing as if vector genomes are like centimeters or milliliters. They're not the same, even though they're called vector genomes. They're very specific to the transgene of interest that you derive that material for. It's not the same target. It's not the same affinity of the primer probe set to that target. So I really don't know what 1E14 vector genomes dosed in in Zolgensma would mean in terms of efficacy, or whether it's high or low, as compared to 1E15 vector genomes for another neurological disease. It does not necessarily mean that those two numbers are 10x different or they are the same. There's no way to compare them because it's an apples and oranges kind of conversation. And it helped her so much to kind of think about it, because she had always thought of vector genomes as being something absolute, as if you're talking
Inviolate.
in milliliters or in another unit of measurement, right? So I think that's a great point to make.
Yeah, for me, I think the disconnect often is all teams have to think in terms of, let's say it's a fusion of vector genomes plus empty full capsid, right? Like, because that number in your head becomes your immunogenicity risk for whatever you're delivering. Correct. Right? But on the vector, like the raw vector genome data, like it's important for within your product and maybe between your products to know how to scale your platform, your manufacturing platform. But it's really, really impossible to compare apples to oranges between products on that. Yeah. Okay, so we're at the end of our time, and I have my favorite final question for you, which is, if you could wave a wand and fix one thing about gene therapy development, what would it be?
So my
and you can have more than one. You can have more than one if you need.
Okay. Mine is a little bit too philosophical, I feel, and I think it impacted me hugely when I was in the therapeutics company. We were working with ultra rare disease in Battens, and were unfortunately unable to take it through post CMC. And it really bothered me that we weren't able to help those patients. And so I think a large part is the cost of a therapeutic that we make. I understand they're one and done. I understand that we're thinking about it being paid right away versus the standard of care that goes on for through the lifetime of that patient would probably be more than what the cost of a therapeutic is that we're talking about. But nevertheless, it feels like it's impossible for a lot of diseases. And sometimes it's impossible to actually commercialize a product because the patient population is so small. So, and I understand also that a large part of that cost comes from the cost of goods of manufacturing. So especially for AAV, I feel like having those several different serotypes forces us to get bespoke and build processes that are really unique for each product, and it adds to that high cost. So my magic wand is going to actually figure out how we can make the whole paradigm simple. Change the paradigm, make it simple, make it agnostic, find a one-size-fits-all with the options to tweak and optimize rather than to completely overhaul and make bespoke. I think that would be a huge success. But like I said, it's very philosophical because it's not one thing that changes. It's multiple little things that need to change to make that work.
Yeah, no, that resonates very, very strongly. I think that figuring out how to increasingly platformize, to use your word, what we're doing so that we can drive down cost of goods. We can also, frankly, increase our knowledge base, holistically. It still won't be apples to apples comparisons, but it'll be much closer to that. And that could very well get these therapies to more patients, especially those in ultra rare diseases, for sure. Well, Susan, thank you so much for joining us on the issue. Really appreciate you spending the time and sharing your knowledge. This was a lot of dense knowledge that you've shared with us, and I hope people enjoy it. I certainly enjoyed our conversation.
Thank you, Emily. Thank you for having me on the issue. I really enjoyed our conversation as well. Thank you.